Hi, I'm Lawrence Patterson. I'm Professor of Drug Discovery and Director of the Institute of Cancer Therapeutics here at the University of Bradford. I can sum up what we do at the Institute in a mission statement. We develop new cancer therapies from concept to clinic. In doing that, we look at a variety of targets that we're interested in developing new drugs towards. Those targets are proteins and DNA that are associated with the cancer process. The experiment you're participating in today is all about DNA, so straight away you can see what the connectivity is. You're actually going to be looking at the effects of chemicals on DNA, chemicals actually you wouldn't want to normally interact with DNA. For example, when somebody that's a smoker smokes, they are directly damaging their DNA. But actually for us at the Institute, we are interested in deliberately damaging DNA using drugs. And in fact, many of the drugs presently used in the clinic today deliberately damage DNA. So you can see there is a sort of a connectivity in that regard. You will understand your own DNA, you will look at molecules that combine to DNA, and I hope you will get some flavour of the sort of work that we're interested in, as well as learning how to look after your own DNA.
DNA has been a problem as a target for some years now. We are developing prodrugs that can be metabolized selectively in tumor tissue, and the metabolites will then damage DNA and hopefully stop the tumor progression. In terms of measuring the DNA or the damage to DNA by these new small molecules, there are various ways we can do this. At this institute here, we are looking at doing that using two different technologies. One is using DNA sequencing. DNA sequencing is a very powerful way of detecting DNA itself, but also damage that has occurred to DNA. We can do that by using equipment like what I've got here on the left, which is a tank that you can use to run a sequence of DNA material that has been incubated, for example, with a drug. Then once it's been running on the gel for a couple of hours, we can detect what has happened to the DNA. Another experiment you can use to measure the DNA binding of a new ligand, such as a drug molecule, is to use DNA thermal denaturation experiment. We do at the equipment we have got at the back here. Basically what happens is that you incubate the ligand with the DNA, and you incubate it for either minutes or up to several hours, and then you detect how the DNA has been stabilized by the binding of the drug molecule. These are some of the two techniques we use at the Institute here, but if you want to have other sort of information from the binding of the DNA, we work with collaborators outside the Institute.
Within our bodies there are organs and tissues that are composed of small packages called cells. And within each of these cells is a remarkable molecule called DNA. Now, DNA controls many of the features that make us who we are: hair color, eye color, etc. Very important molecule, controls our life. It's really important to take care of your DNA, because if you damage it, it can lead to certain diseases, and particularly diseases such as cancer. In this particular practical, what we're going to do is to extract some DNA from a mouthwash and have a look at it. We're also going to show you just how easy it is to damage the DNA by exposing it to chemicals that will bind to the DNA. The first step in the procedure is to obtain some cells, and then what he's going to do is to have a mouthwash and swirl a solution around in his mouth for about 30 seconds, and that's going to take some of the cells off the lining of his mouth, and then he's going to collect those in a tube. So the next step in the procedure is to add that mouthwash to another tube that contains a small amount of detergent. This is like a washing up liquid that he's going to use, and that's the purpose of the detergent is simply to break open the cells and allow the DNA to come out of the nucleus of the cell. Okay, it takes a few minutes for the cells to break, and during that period he's just gently going to rock the tube up and down, side to side. Okay, the final phase of the extraction procedure now is to add some very cold ethanol or alcohol to the mix. And what Toby's going to do is add this very carefully so that the two solutions don't mix. So what he's doing is just gently layering it down the tube, and the alcohol is just going to sit on top of the detergent solution that he had before. The DNA will come out of solution and precipitate and form a white stringy mass at the interface between the ethanol and the detergent. Okay, so what Toby's doing now is collecting the DNA from the tube, and then he's going to put it into another tube that contains a chemical, and this chemical is something that we've isolated from cigarettes. This particular chemical will bind to the DNA. It only takes a few seconds to bind to the DNA. And when we put it under a certain light source, what you will see is the DNA glowing. Okay. And that represents the actual chemicals extracted from cigarettes that are binding to DNA. Okay. It clearly indicates that these chemicals have bound and they've stuck to the DNA. And that process of sticking, or chemicals sticking to DNA, causes mutations in the DNA. It damages and changes the DNA, and that can lead to diseases. Okay, this practical session is going to take place on the 12th of September between 10 o'clock and 12 o'clock in the atrium at the University of Bradford. So we look forward to seeing you there.